Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Edexcel International A-Level Biology Unit 2 for October 2022. Let us begin with the first question. Question 1 said, Wallace classified organisms into a system with 3 domains. How many of these domains contain prokaryotic organisms? These are 2. We have the archaea as well as the bacteria. Then the next part says, the diagram shows part of a prokaryotic cell as drawn by a student. So they've labeled for us the pili, and they say complete the diagram by drawing and labeling the flagellum and a plasmid. Flagellum, we can see that, and then the plasmid, I put it inside, which is going to be labeled there as well. The next part says, state the function of a plasmid. A plasmid contains genetic material that is used for synthesis of proteins. Here they say the ribosomes in prokaryotic cells are different from the ribosomes in eukaryotic cells. Give one difference between prokaryotic ribosomes and eukaryotic ribosomes. Now, if you remember, eukaryotic ribosomes are found on the rough endoplasmic reticulum attached onto the membranes, while the others are not. Another thing is, in prokaryotic, we have the 70S, while in the eukaryotic, we have the 80S. So I said prokaryotic ribosomes are 70S, while eukaryotic ribosomes are 80S. Also, prokaryotes have free-floating ribosomes, but eukaryotes have ribosomes attached to membranes. Then they say how many of the following statements about eukaryotic ribosomes are correct. Ribosomes are located on the surface of both types of endoplasmic reticulum. This is wrong. They are only found on the rough endoplasmic reticulum and not smooth. Ribosomes are located inside chloroplast and mitochondria. That's true. And ribosomes are involved in the process of transcription. That is wrong, because transcription occurs inside the nucleus. They are only involved in translation. So the answer here is B. Only one is correct. So this brings us to the end of question 1. Let's continue to question 2. Question 2. The photograph shows water lilies in a river in Thailand. One water lily leaf has a diameter of 305 centimeters, which is about 3.05 meters. They say which of the following shows the upper surface area of this leaf? Assume the leaf is circular. Since we have the diameter, radius is going to be diameter divided by 2, which is 152.5 centimeters. And then the area is going to be pi r squared if we assume it's a circle, which is going to be pi into that radius squared, and it gave me this. But we can see this is in centimeters, and it's not exactly that. So I try to convert this to meters squared to see if the answers in meters squared correspond to this. 1 meter is equal to 100 centimeters, and 1 meter squared is going to be 10,000 centimeters squared. So dividing by 10,000, it gave me 7.3, which is what we see here, 7.3 meters squared, and the answer here was A. Moving on. Part B says the photograph shows a cross-section of the stem of a water lily. So they say the structure labeled X contains xylem vessels and phloem. How many of the following statements are correct? Both xylem vessels and phloem transport dissolved substances. That is correct. The xylem transport dissolved minerals, the phloem transport dissolved nutrients. And then only phloem transport dissolved substances in one direction. That is wrong. Only the walls of the xylem vessels contain both cellulose and lignin. That's true. So 2 correct things, and therefore the answer is going to be a C. Moving on. The leaves of the water lily contain several types of tissues and cells. The photograph shows a group of leaf mesophyll cells as seen using an electron microscope. Label 1 permanent vacuole on this photograph. This is the permanent vacuole. Give 2 functions of a vacuole. A vacuole stores dissolved substances. It will store proteins, it will store mineral ions, as well as enzymes and sugars. And then it provides support by maintaining osmotic pressure to ensure that the cell remains turgid. So this is the end of question 2. Let us continue to question 3. Question 3. The photographs show plants growing in vertical and horizontal systems. In both of these systems, the plants grow in nutrient solution instead of in soil. So we can see this is in vertical and that is in horizontal. The nutrient solutions contain magnesium ions and nitrate. Describe the roles of these inorganic ions in the plant. Magnesium ions, of course, are useful for making chlorophyll so that the plant can trap more sunlight during the process of photosynthesis. while nitrates are useful in making amino acids as well as nucleotides. Then they suggest one advantage of the vertical system over the horizontal system. The vertical system is going to save up on space. As you can see, a lot of horizontal space is used here, but if they were all vertical, more plants could be grown within that small space. So I said more plants can be grown in a given space, thus increasing the yield as well as the profit. Moving on. In part B, fruits contain cells. The effect of calcium ion concentration on the firmness of fruit was investigated. Cherry plants were treated once, twice or 3 times with 2 different concentrations of calcium chloride solution. The firmness of the cherry fruit was then measured. The table shows the effect of these treatments and a control. So we have the number of treatments from 0 to 3, plants treated with a calcium chloride solution, which is lower, and those treated with a higher. This is actually 4 times higher. So we can see overall, those treated with 950, there is going to be an increase in the firmness. And here we can see an increase in the firmness, but this is higher in comparison to that. So they say, which is the percentage increase in mean fruit firmness between the cherries from the control plants and those from the plants treated 3 times with a higher calcium chloride concentration? So considering that, which is the higher treatment, the increase is going to be that, and so the percentage increase is going to be that, divide by that, times 100, which is 14.39, and the answer is going to be a D. Moving on. They say give one conclusion for this investigation. Of course, increasing the number of treatments increases the fruit firmness, as we have seen in the table. The values are increasing with more treatments. And here they say explain the effect of calcium ion concentration on the firmness of cherry fruit. Remember, calcium pectate is made out of calcium, and that is found in the middle lamellar, where it holds the cells firmly together. Every time they ask you to explain, write a statement and then give a reason as to why that statement is the way it is. So I said, as calcium ion concentration increases, fruit firmness increases. Calcium ions are used to make calcium pectate, which is part of the middle lamellar. So a higher concentration of calcium pectate in the middle lamellar leads to more cells being firmly held together, so fruit firmness is increased. So this brings us to the end of question 3. Let's continue to question 4. Question 4. Boards used for chopping fruits and vegetables can be made from wood and oil-based plastics. So they are here. Some are made out of wood and others are made out of plastic. They say bacteria can grow on the surfaces of chopping boards. Explain the conditions that would result in the greatest bacterial growth on the surface of a chopping board. For bacteria to grow, we need a source of carbon, which is a sugar. So if there is presence of glucose or a sugar, if there is presence of the right pH, as well as conditions that allow bacteria to carry out respiration. So I said presence of a carbon source or sugar like glucose and presence of oxygen can allow for aerobic respiration to occur and ATP to be generated. The optimum temperature can speed up the enzyme-controlled metabolic reactions, and then the optimum pH can allow for faster rates of enzyme-controlled reactions, and also presence of water. This helps in hydrolysis reactions to occur, and water acts as a solvent to dissolve the metabolites for faster diffusion. Because remember, bacteria release enzymes that carry out external digestion, and then the nutrients have to be absorbed. So those nutrients have to be able to dissolve in a specific solvent so that they can be absorbed back into the cell. So moving on. Explain why boards made from oil-based plastics are not considered sustainable. To be sustainable, it means they're renewable, but in this case, those made from plastics are made from non-renewable resources. They are not biodegradable, so their waste is going to accumulate, and also because they contribute to the release of carbon dioxide, which is a greenhouse gas. In part B, the number of bacteria surviving on boards made from different materials was investigated. Identical bacterial cells were placed on boards made of pine wood, spruce wood, and plastic. The graph shows the number of bacteria surviving after bacteria were placed on the boards and kept in the same conditions. So we can see this is 1 hour, and that is 4 hours and 24 hours. Here we have the lowest number of bacteria surviving in each area at all times. Then we go to spruce. In spruce, initially, or I would say 1 hour, spruce as well as plastic had the same concentration or same number of bacteria, but at the 4th hour mark, those on spruce had decreased while those on plastic were still the same. And then at 24 hours, we saw a decrease. So here they say, comment on the results of this investigation. So I said overall, the number of bacteria surviving decreases over time on all boards. That's true. They all decrease. And then pine chopping boards had the lowest surviving bacteria per area at all times. And bacteria survive the longest on plastic chopping boards. There is no change in the number of surviving bacteria at 1 and 4 hours on plastic chopping boards. That is true. We have it here. And then there is no evidence that the experiments were repeated per condition, no mean or standard deviation, so the results are not valid. And again, here they talked about commenting, so I have to comment about the validity of the results as well. And down here they say, suggest one reason for the difference in bacteria survival on the pine chopping boards and on the plastic chopping boards. The reason as to why there were less bacteria surviving on pines is because maybe they have antimicrobial properties. So I said pine boards have antimicrobial properties, and pine boards may not provide the optimum conditions like the right pH, so the growth was lower, or there was less growth. So this brings us to the end of question 4. Let's continue to question 5. Question 5. Some of the characteristics of an organism are due to polygenic inheritance. Every person has a rhesus characteristic. People who are rhesus positive have one or more of 3 types of rhesus antigen. There is C, D, or E on the cell surface membrane of their red blood cells. Each rhesus antigen is coded by a dominant allele of gene C or gene D or gene E. People who are rhesus negative for one of these genes will have 2 recessive alleles. For example, a person who is rhesus D negative will have inherited 2 recessive D alleles. The diagram shows the loci of the 3 genes on a pair of chromosomes. So they say state what is meant by the term allele. Allele is the alternative or version of a gene. Next they say, which of the following is the genotype of a person who is rhesus negative for all 3? Remember, rhesus negative has 2 recessive alleles. So if they are rhesus negative for all 3, it means they have small c, small d, and small e. So A is going to be my answer. Next they say, rhesus antigens are glycoproteins. Describe how a rhesus antigen will be produced from its polypeptide chain and transported to the cell surface membrane. This question is about from the process of making the polypeptide chain to the point when this polypeptide chain is transported to the cell surface membrane. So we are talking about ribosomes, talking about the rough endoplasmic reticulum, the Golgi apparatus, until packaged in the vesicles. So I said, after translation, the polypeptide chain leaves the ribosome and enters the rough endoplasmic reticulum. In the rough endoplasmic reticulum, the polypeptide chain folds into the secondary or tertiary structure. The folded protein is packaged into transport vesicles and it is transported to the Golgi apparatus. In the Golgi apparatus, carbohydrate groups are added and now the glycoprotein is formed, and the glycoprotein is packaged into secretory vesicles. These are transported to the cell membrane. Moving on. In part B, seed color in wheat is an example of polygenic inheritance. There are 3 genes thought to affect seed color. There is gene A, gene B, and gene C. Each gene has a dominant and recessive allele. A cross between 2 heterozygous plants, which are those, was carried out, and the seed color of the offspring was recorded. The table shows the phenotype of the wheat seed color and the number of recessive alleles and the frequency of the seed color in the offspring. We see as the number of recessive alleles decrease from 6 to 0, the color becomes darker. And here we see the frequency. This one has the highest frequency, the intermediate rate. So here they say explain this variation in color of the wheat seed. And again, you have to know it's an increase in the darkness of the color as the number of recessive alleles decrease. So here I said the higher the number of recessive alleles, the lighter the color. And this is going to be due to variation in gametes that is caused by either random assortment and crossing over during meiosis 1. Fertilization is also a random process, so it can lead to offsprings that have variation in color. So fertilization occurs randomly, and this leads to great variation among offsprings. So I say dark red has 0 recessive alleles because there was a random fertilization that occurred between gametes with no recessive allele at all. So this brings us to the end of question 5. Let's continue to question 6. Question 6. The diagram shows some of the steps involved in the production of human sperm cells from an unspecialized cell. The process involves both mitosis and meiosis. So this is the structure we see here. Next, the unspecialized cell is diploid. State the type of cell division occurring in step A and step B. In step A, because the result is still 46 chromosomes, this is going to be mitosis. And here we produce haploid, so that is going to be meiosis. So mitosis and meiosis. Next, they say which part of the sperm contains enzymes that are released to break down the zona pellucida. This is going to be the acrosome, and that is going to be part W, as you can see. So my answer is A. Next, they say a sperm cell is a specialized cell. Explain what happens at step D for the cell to become a specialized cell. Let us see here. This cell looks differently, but it changes to look like this. It means there is going to be epigenetic control. The cell is going to differentiate in order to become a sperm cell. So I said epigenetic modification like DNA methylation as well as histone modification leads to gene expression. So some genes are switched on while others are switched off. The expressed genes are transcribed into messenger RNA, and then the polypeptides or proteins are formed, and these formed proteins will change the structure as well as the function of the cell, meaning the cell becomes specialized in order to change the structure, the shape, as well as the function. Moving on. In part C, the nucleus of a sperm cell fuses with the nucleus of an egg cell to form a zygote. The zygote develops into a blastocyst. They want you to draw a diagram to show the arrangement of cells in a blastocyst. It's easy. Just draw a line of cells around and then a cell mass that has to be put inside. There should be some void area. That is what they're looking for. Next, they say the blastocyst contains some pluripotent stem cells. Which of the following describes these pluripotent stem cells? A says adult stem cells that can give rise to most of the cell types in the body. Because they said blastocyst, these are not adult stem cells. So anything that has adult is out. So they say embryonic stem cells that can give rise to most cell types in the body, including totipotent. They never give rise to totipotent, so that is wrong. And then embryonic stem cells that can give rise to most cells in the body, excluding the totipotent, that is right. So D is the answer. Moving on. Here they say the photograph shows a Mexican salamander. Salamanders can regenerate damaged limbs and organs throughout their lives. An ethics committee has approved the use of stem cells taken from salamanders, suggests why the use of stem cells from salamander embryos was approved. If they're approved, it means the salamander embryos do not have fully developed nervous systems, so they will not feel the pain. Also, stem cells taken from salamanders can differentiate to form specialized cells in tissues as well as organs. When we use salamander embryos, it means the need to use human embryos can be dealt away with. And also, the potential benefits outweigh the disadvantages. So this brings us to the end of question 6. Let's continue to question 7. Question 7. Great Lakes and Crater Lakes can be found in Nicaragua, a country in Central America. These two types of lakes differ in their species richness. State what is meant by the term species richness. This is the number of species within a specific habitat. In part B, they say Lake Nicaragua and Lake Managua are Great Lakes. Crater lakes are formed from craters of volcanoes. The photograph shows a Great Lake and a crater lake in Nicaragua. So when we go down here, they say the diagram shows a proposed relationship. So we can see the crater, we have that as well as that, and then we see those that are in Lake Apoyo, and then those that are in the Greater Lakes and so on. So they're different species because there is some speciation that has occurred. So also they show us the location of these lakes. We have the Lake Managua as well as the Lake Nicaragua here. And then we have the two crater lakes, Lake Shilowa as well as Lake Apoyo. So they also give us extra information about when these legs were formed. For the crater legs, Apoyo was formed 400 years earlier. And here we can see there is a 1,000-year difference, while here we have 400-year difference between their formations. So here they ask, describe how new cichlid species may have been formed in these four legs and how the scientists would have determined that these cichlids were 6 different species. Use your information in the question and your own knowledge to support your answer. So, and again, we are talking about formation, so we have to go back to look at the map. We can see there is landmass here. Something could have happened that could have caused isolation of the cichlids initially to create cichlids that are in different habitats. And then over time, as crater lakes were created, some of the other cichlids were trapped into specific areas, and due to that geographical isolation, speciation resulted. So I said, initially, cichlids were isolated in different habitats of the same lake, although cichlids could still move between the Great Lakes. Then geographical isolation occurred between the Great Lakes, and the crater lakes were created. Now from the table, we can see this was formed 4,700 years ago, and here 4,300 years ago. So we can see this was formed 400 years before this was formed. So Lake Apoyo was isolated and inhabited by the cichlid 400 years before Lake Shiloah was inhabited. So mutations occurred and new alleles led to genetic variation in the different habitats. So due to the different selection pressures in each habitat, organisms with advantageous alleles had a selective advantage, and so they survived, reproduced, and passed on their alleles to their offsprings, and the allele frequency increased over time. Also, speciation occurred in Lake Apoyo 1,000 years before Lake Shiloah. We can see it from the table here, 1,000 years earlier. The difference in time is 1,000 years. Now, the last question was, how were these species determined? How did scientists find out that these were different species? Of course, this is going to be either looking at them physically, which is morphological, as well as carrying out molecular phylogeny or examining their DNA. So here I said, to determine if these species are different, analysis of the fish phenotype for similarities and differences in body features and the ability to produce fertile offsprings. Also, biological molecules like proteins and nucleic acids were analyzed using molecular phylogeny to observe similarities as well as differences in order to make conclusions on whether they are the same species or they have evolved to become different species. Moving on. So next they say, a study compared the diversity of cichlid species in 2 crater lakes. The index of diversity D for Lake Shiloah was 2.8. The table shows the data obtained from Lake Apoyo. So as we can see, this is n into n minus 1. So we have, this is going to be 156 times 155, and this is going to be 17 times 16. So this is going to be 12 times 11, which is 132. The sum of this is adding up everything here, and we get that. So down here to calculate the diversity index, an index of diversity is calculated using this formula. So determine which of the lakes has the higher biodiversity of cichlids and use the table and the formula to help you. So we already have this sum. So all I did is substitute. Since we have that, which is that, now this is going to be 316 times 315, which we have here, divide by the answer we get here, and I got 3.05 as my answer. So here I said, Lake Apoyo has biodiversity of 3.05, which is higher than the 2.8 of Lake Shiloah. Moving on. The drawing shows 2 citrinellars cichlids. There is one that is melanic and the other that is gold. So they say the gold phenotype is caused by inheriting a dominant allele, and the melanic phenotype is caused by 2 recessive alleles at the same locus. A scientist studied the change in allele frequency in the A. citrinellars population over time. In the first study, the allele frequencies in the population were P is equal to 0.6 and Q is equal to 0.4. In a later study, 672 fish of this species were gold and 128 were melanic. Determine if the allele frequencies have changed using the equation P squared plus 2PQ plus Q squared is equal to 1 and P plus Q is equal to 1. These were gold and these were melanic. The total is going to be that plus that. So to determine the melanic, which is recessive, I said melanic Q squared is equal to 128 divided by 672 plus 128, which gave us Q squared. And therefore Q is going to be 0.4. This is the allele frequency for the recessive phenotype. I use the formula P plus Q is equal to 1. So P is 1 minus Q, which is 1 minus 0.4, which gave me 0.6. So we can see the allele frequency is still Q is equal to 0.4 and P is equal to 0.6. So allele frequency has not changed. So this brings us to the end of question 7. Let's continue to question 8. Question 8. Mitosis and the cell cycle are involved in producing new cells. Describe the role of the cell cycle in producing new cells. So the cell cycle contributes to DNA synthesis during replication. Also, there is synthesis of new organelles that are going to be used to make new cells. Also, after cell division, it allows for the cells to grow and increase in size during the interphase stage. So those are the 3 answers I wrote. Moving on. Here they say, uncontrolled cell division can result in the formation of a tumor. The RB gene is a tumor suppressor gene, meaning it suppresses the formation of tumors. So they say the RB protein produced from this gene inhibits tumor growth. Then cancer can occur when this gene has increased methylation compared with the RB gene in normal lung cells. Explain why increased methylation of this gene could cause the growth of a tumor. We know that if methylation occurs, methyl groups can be added to either DNA or onto histones, preventing the transcription of a specific gene or causing the silencing of a gene. And that means RNA polymerase as well as transcription factors cannot bind to cause transcription of that gene, and meaning the protein will not be formed. So I said, during increased methylation, more methyl groups are added to the DNA or histones, so the gene will be switched off. So RNA polymerase enzyme and transcription factors can bind to the gene. So transcription of the gene and translation of the messenger RNA into the tumor suppressor protein can't occur. So mitosis continues without inhibition of tumor growth. Moving on. In part C, the cell cycle can be affected by various chemicals. One drug used to treat various types of cancer prevents the shortening of the spindle fibers. Explain how preventing the shortening of the spindle fibers affects mitosis. Shortening of the spindle fibers allows for the sister chromatids to be separated. So if that doesn't occur, then it means they will not be separated. So failure to shorten the spindle fibers results in failure to separate sister chromatids, as the centromere will not split during anaphase. Moving on. Paclitaxel is a drug used to treat some tumors. In one study, the effect of paclitaxel on the cell cycle in human lung tumors was investigated. The table shows the results of this study. We can see as we increase the concentration, duration in mitosis increases, and then the mitotic index of cancer cells in the lungs also increases. It means mitosis is taking longer, and now there are more cells in mitosis. Because if mitosis was faster, it means cells are going to be speeding up. But if there is more cells in mitosis, in this case, it's taking longer, and therefore the drug could be effective. So they say in another study, the effect of paclitaxel on the growth of breast tumors in mice was investigated, and the effect of paclitaxel was compared with drug X and with a placebo. For each group, the increase in mass of the tumor was measured 21 days after starting treatment. 3 groups of mice with breast tumors were given the following treatments. Group 1 received a placebo, Group 2 received drug X, and Group 3 received paclitaxel. The graph shows the results from this study. The placebo, the mean mass of the tumor was huge. With drug X, we can see the mean mass of the tumor is large, but it's not as big as that in the placebo group. And for paclitaxel, we see the mean mass of the tumor was lower, and that means the drug could be working. Also, when we look at the error bars that have been put, there is overlap here, so these 2 results may not be reliable, but because there is no overlap between this and the rest, we can see the results could be dependable. So here they say, evaluate the effectiveness of paclitaxel. Use the information given to support your answer. Now, my first points went to the table and then the next went to the graph. So from the table, the higher the concentration of paclitaxel, the longer the duration of mitosis. We can see from 1 hour at 0 nanomole to 24 hours at 10,000 nanomole, we can see there is increase in the duration and there is increase in cells that are going through mitosis. So from the table, the higher the concentration of paclitaxel, the higher the mitotic index of cancerous cells in the lung tissues. It increased from 2% at 0 nanomole to 59% at 10,000 nanomole. Then from the graph, paclitaxel causes the lowest increase in mass of the tumor, so it is the most effective compared to drug X as well as the placebo. Also, presence of error bar shows variability in the data. Lack of overlap between paclitaxel and the other two drugs shows existence of a significant difference, so results are valid. We can also see paclitaxel decreased cell division of cancer cells, so less tumor mass resulted. Increased duration of mitosis led to increased mitotic index. This is explaining we can see there was an increase in the number of cells going through mitosis because spindle fibers are not separating and therefore many cells are left in anaphase. It doesn't mean that there is more tumor cells that are dividing. It just means there are more cells that are not going into the next step of mitosis because there is prevention of spindle fibers from shortening. So this brings us to the end of question 8 as well as to the end of this paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.